All right, so I want to make this quick, but also, to address a couple questions that people might have about the summative. And I'm going to try to start it with the things that most people might want to know, and then I'll do some of the individual presentation of the results, like the different groups later on, so that you can kind of just click through and get to the part that was about your own data and things like that. Okay, so just really quickly first, on the unit plan, I just want to go back to like some things that might be helpful. So this is the PowerPoint. It's actually about all things having to do with plants. So you probably only really want to look at a certain part of it. And I especially wanted to highlight this. So this is a sample long answer that would normally be worth 8 points, but notice that this answer key, the stuff that I took off is about mostly about how the roots absorb water. And I know that some people ended up looking at this anyway as they were looking through the chapter, but we really haven't learned about active transport or osmosis. And so this is going to be a lot easier to understand after that. So I just wanted to let people know that that is not really an expectation, that you're going to know all the details about that now. But that's going to be really easy to go back, and once we learn about these things, it'll be really easy to explain how those are used in the transport of water into the roots, which helps set up some of this pressure difference that's going to move water upwards. But then you also have the option to look at the second part of this key, just to kind of look at some of the terminology that would be good to include in your answer. And so again, this is what an answer key might look like. It's not always going to be exactly like this. So you want to look as you're studying for, terminology that would help you explain these things, the connections between differences in pressure, this like pressure potential, but this difference in pressure that's going to move fluids from the bottom to the top of the tree. Okay, so there's lots of other things in here too, but just like this is super helpful to kind of have an idea of what's going on in the leaf. Not too much about guard cells, so I wouldn't focus on this too much. The structure of the xylem and lignin, just essentially maintaining the strength. And then again, roots, like don't worry about this too much right now, so I wouldn't worry about this one too much. Again, don't worry about that. But this is a helpful thing to take a look at. Adaptations or, structural feature of xerophytes are plants that live in dry habitats like the desert. And so, you can probably relate to this thick waxy cuticle or small thick leaves like like think about cacti or things that don't really have a lot of surface area for losing water compared to leaves on a lot of other plants. And then this would be an example of like the underside of a leaf being rolled up and having stomata inside of these pits, so that the more humid this is, the less water, the more humidity that's trapped in here from the evaporation of water will lead to less transpiration. And then there's this won't probably make quite as much sense at the moment because we haven't talked about, so I wouldn't worry too much about this, but kind of some similar things. But these are going to be plants that are living in really like salty soils, and so I wouldn't worry too much about this right now. Okay, so let's look at some of your transpiration results. So I put everything into this one, and then I wanted to just really quickly like click through some of the things you were looking for. So explaining the question, and some of the things that you're going to see are going to be questions that, anybody who did the experiment should have an idea of, how do we calculate a transpiration rate? There are going to be some kind of easier things based on whether you did this. And then maybe there will be some more advanced questions that you can kind of show, what's your depth of thought about these and experimental design that would be different. So just looking at all of them at first, so I just want to go through some kind of trends that I saw, so everybody should have an idea about independent variables, dependent. The interesting thing about the controlled variables was that, people tended to list about 3 things. Although let's kind of click through here. I don't know that anybody was able to control their surface area, but you might have said something about trying to pick plants with, similar amounts of leaves, but you probably couldn't control for that. So you might phrase this in another way. You didn't control for the surface area, but you could have mentioned, picking plants with a similar amount of leaves, or I guess you could explain that, that you tried to control surface area, but you knew it wasn't exactly the same. Yeah, type of plant. And then actually it's interesting because this group pointed out that their light intensity might not have been controlled. All right, so let me just look through one thing that I thought was pretty good about, yeah, so this group mentioned the area. So you can think about not only the area, but what were the conditions where you took your sample from, what was the same about that condition, and that ideally, there are multiple things that you would want to control about the place that you're taking your samples from. um, yeah, similar idea, but yeah, I think you can think about like what was the same about your plant, your sample, and the conditions that it was grown in. So hopefully people have an idea about controlled variables. But this is interesting. So for this group, the distance from the fan actually was not controlled because that was their independent variable, right? One group was, but I guess they're trying to say that for the ones in the fan group, the distance was the same. But yeah, again, it's interesting, like amount of water and oil probably wasn't controlled. Again, surface area probably wasn't controlled, but you could mention trying to control it. But this one, actually, this group pointed out that their surface area wasn't the same between different plants. So you'd want to think about what did you try to control, but also in the evaluation, what you should talk about is the variables that weren't controlled, but you very well could have controlled them. And so again, if you were going to do this again, what would be the variables that you would control in order to set up a well-controlled study? All right, so the one thing I really wanted to look at quickly was the graphs of the results in comparison between the different groups. So keep in mind that you ideally want at least one graph that will answer your research question. And so one of the biggest things that I saw for the graphs was like some people put their two conditions on different graphs, and in some cases with different scales for the y-axis. So if your question is about lamp and no lamp, so this is one of the ones that I thought did a better job of comparing them, and some of this might just be people not being totally comfortable with graphing in Google Sheets and knowing how to do that. But this one you can really clearly see, these are the 5 repeats for the lamp, these are the 5 repeats for the no lamp, and how do those compare? And it was interesting, this group, I think sort of accidentally, these were lined up mostly from highest transpiration rate to lowest. But think about, your trial 1, 2, 3, 4, 5, you could put these on a graph from highest to lowest. And so the thing about this group, if you look at almost every single lamp had a higher transpiration rate than no lamp, except for this last one. And so that, will give you an idea that, probably the ones in the lamp, that condition has a higher rate of transpiration. But this graph, I thought, did a really good job of comparing them. The other one that I thought was, this one, right? So you've got, here are the 5 samples for shade, here are the 5 samples for the light. And then eventually we're going to get to the place where you have something like this. So comparing the two averages. And then, again, some people don't know how to do this yet, but looking at the, some kind of a variation in those data points. And you can see that here there's a lot of variation in their shade, because they had these two data points that had really high rates of transpiration, where all the rest were down in this really low, low category. But, for some of the other ones, like this one was hard. So you had the no flowers and flowers, but it's just hard to compare also because they're on two different graphs and they don't have the same y-axis. And then this one too, there were some graphs without the fan and there were some graphs with a fan, but really, really difficult to compare them to one another. So that will give you an idea of kind of what kind of graph. The other thing that a lot of people had, making sure that these titles are succinct, what were you measuring and the units, obviously that would ideally be present on a graph that somebody could tell what you were looking at there. Okay, so the conclusion, again, in some cases it was hard to say for something like this. It would be easier to make a conclusion. I wouldn't worry about that too much, except there are definitely people that, were able to say, yeah, like it's really hard to say, were these high rates of transpiration telling us something, or were they just sort of, well, a reflection of the fact that there weren't that many leaves. So mostly what people would have been able to get out of these is what would you do differently next time? But I'm not sure if these, let's see, actually, yeah, these guys probably were able to come to a conclusion too, what they say. So they said no noticeable difference, although interesting. So these are zero point 0.1. Yeah, there's no noticeable difference between those. But notice like kind of you really, really have to look at the axis to tell if there's a difference. Okay, so the scientific context is where you're going to try to explain the results. And I was looking at, I'm just kind of trying to flip through here. So the lamp and the no lamp, um, people tried to reference, and I know some other groups did this too, would stomata be open or not? That might affect the rate of transpiration. So this group looked at if there are lights, maybe that will result in the stomata being open, and there will be a higher loss of water. But then also this. So we talked a little bit about light changing temperature, but keep in mind that light is a form of energy as well. So radiant energy. You could talk a little bit more here about what is that energy being used for. And when you think about transpiration, we're talking about evaporation, and we know that that takes energy. So you could even talk about, energy breaking the hydrogen bonds to, cause water molecules to turn into vapor, vaporize, that latent heat of vaporization. You could talk about that a little bit more in scientific terms. Why would light have an effect on transpiration other than by increasing the temperature? But there are certain things where, yeah, like temperature, humidity can have an effect on transpiration rates. And then this is interesting down here, like a fan can also have an effect. So you would expect that removing water molecule or like moving the water vapor in the air by a leaf, it will actually cause evaporation rates to increase. So trying to address your explanation here. And then there's a lot that people can look at in terms of evaluation. So let me see if I can go through a little bit with some examples here. I'm trying to remember where I can pull from. Okay, so yeah, I would think for strengths, often they fall into two categories. One is about controlled variables. How well did you control the variable? And then the other thing that I kind of saw consistently referenced was sample size. So let's look here, right? So these talked about controlling, that's a good strength. Accounting for the different surface areas. So even though you couldn't control for it, factoring that in so that you could actually compare the different plants. That's a really good strength. Let's look for, and I might be missing some things here, but I'm not sure. Did these guys have strengths? So they, I think, started with improvements, and then I want to see what, I think there were some up here. And then I think we need to be more specific about what was strong about the method. But here's the other thing, right? Like how many, the fact that you had 5 in each group, a lot of people mentioned that you could increase the number of plants, right? Maybe 5. So like this one, maybe 5 wasn't enough to get rid of some outliers. Like maybe you really would have wanted to see what happened with 10 in each condition. But there were some interesting, and here's where the creativity, well, I'm curious what this means, small number of recorded values, but you could say like, we would want to see what happened over a 10-hour period of time instead of just 3 hours. Let's see what else here. Yeah, these guys had a specific idea about, the plants might have been getting different levels of light. Let's look really quickly. Yeah, these guys had a looking at getting plants with a certain surface area. Let's see what these guys. You could talk about how you could better control variables. Right, they had a specific one about measuring the surface area before the leaves kind of dried up. Let me look at this last one because they had some here. Um, so I think you want to, and I think you just want to be careful about saying, like image J, I guess you can mention this, but can you think of a more accurate way to measure the surface area? I honestly can't. So, I actually think image J is probably a strength. And, the other group mentioned factoring in surface area, but there are other ways to measure surface area that aren't as accurate as image J. Right, so you could have looked at the size of leaves a little bit better. Constant transpiration. Yeah, there's actually a lot of other um, opportunities for improvements here. And here's where the first time that you do this, we were kind of just trying to see where everybody's at. And so, I think the idea is for everybody to kind of see where everybody is and then improve in terms of the creativity and but also the specificity of what are all the ways in which we could have improved this. And so I think I'm actually going to stop this one, but then I might kind of look through the individual ones separately just to see if there are some other things that occur as I'm looking through here.